I was asked to do a short presentation about the mink outbreak and our One Health approach on that in the Netherlands. I was asked to keep it short, so I will do that. I will try to share my screen now. Okay, the mink outbreak in the Netherlands. Well, the work we did on the mink outbreak, and we're still doing a lot of work, is done by a number of people from a number of institutes. So it was a real One Health cooperation to do this research. Main partners in the research were Wageningen University, the Animal Health Service, GD Animal Health Service in the Netherlands, Utrecht University, and Erasmus University Rotterdam. What happened in the Netherlands is that in the end of April of 2020, there was an outbreak. There was clinical respiratory disease observed in mink. And as soon after that started, it was suspected to be a SARS coronavirus infection. So the animals were tested for SARS coronavirus, and it appeared that these animals were indeed infected with SARS coronavirus. After the start of the outbreak, after this was confirmed, we started this One Health consortium that I already talked about. And within a few days, we started necropsies of the animals of the outbreak farms, and we started testing these animals by PCR, both nasopharyngeal swabs and rectal swabs. Within a week, we were able to do full sequencing of the virus, and we immediately submitted that virus to GenBank to make it available to all other researchers in this field. Also because we saw that the virus was very much like the virus seen in humans, and that we assumed that this virus in mink might also be able to infect humans again. So we thought it was important to share the sequence as soon as we could. We also started necropsies in the minks. These are pictures of the first two farms in the outbreak. And actually the clinical picture and the pathology more or less stay the same. The animals showed clear sepsis, lung edema, interstitial pneumonia. And if you would like to see more of this, it's all now available from literature. We have published this paper in veterinary pathology. After the mink outbreak, after it was clear that we had a serious outbreak in mink, transport of mink in the Netherlands was prohibited, and respiratory disease in mink was made notifiable. We started a screening and early warning program in all the farms. So in all the farms in the Netherlands, animals that died were tested for SARS coronavirus every week. In June, the situation was still continuing. There were more outbreak farms, and there was a meeting of the outbreak management, the national outbreak management team, to discuss the situation. And it was decided that we had to start culling all the farms. It was also because that we saw that at first we hoped that the outbreaks would fade out, but during this time, especially in May and June, a lot of young animals were born, and it appeared that these young animals were also infected with SARS coronavirus 2. And that made the situation even more serious. So it was decided that we did have to start culling all the farms. And that was decided by the national government. In June, the situation still continued. In the Netherlands, we had more than 132 mink farms, and the number of outbreaks was still increasing in June. So the culling was continued, and the farm surveillance was intensified. And by the end of June, also the use of personal protection was also intensified. In the beginning, it was advised to wear personal protection in all the infected farms, but by the end of June, people were advised in all farms to wear personal protection, and biosecurity was also intensified. But still, the outbreaks continued. So how did it, what did we see regarding spread of SARS coronavirus in the farms? There were high concentrations of virus in the respiratory tract detected. The number of fatalities in the mink were not so very high, but in all farms there was a clear spread of virus of the infected animals. Rectal swabs were soon negative after the infection, and respiratory swabs always showed higher concentrations of virus. And all the animals, well, at least a large majority of animals in the farms developed SARS coronavirus, SARS coronavirus 2 specific antibodies. The situation became even more serious when we saw that also the people working in the farm were developing respiratory disease due to SARS coronavirus infection. In total, we have tested 97 people working in the farms, 88 by PCR and 75 by serology. This was an overlap of tests. And of the people tested by PCR, 49% were positive, and tested by serology, 51% was positive. So overall, we had 68% of people working in the farms, 66 people diagnosed with SARS coronavirus 2 infection. And of the virus sequences, we obtained 7 full genome sequences, and we could see that the sequences were related to the mink. So it was clear that these people were attracting the virus from the mink. And this paper was a publication in Science. Looking at all the sequences, we could see in the mink farms that there were 5 clusters of sequences. So the assumption is that there were 5 different introductions in the mink farm. And within those clusters, the sequences are related, but also the human sequences were related to those clusters. This is a picture of the geographical situation of the farms. It's all, well, that's the area where the most mink farms are in the Netherlands, or were in the Netherlands. And you can see that the farms are rather concentrated in this area. And in that picture on the right, you can see also the different colors for the different clusters. Cluster A, the red color, was by far the biggest cluster. So since the infections of the farm continued, we were focusing our research, and we're still working on that, to see how the transmission between the farms took place. It was unclear why we couldn't stop the infection between the farms by taking serious hygienic measurements. People were not allowed to come in the farms anymore. All people were wearing personal protection, so they could not spread own infections also to the mink, but still the outbreaks continued, and we didn't know why. So we focused our research, and we're still working on that, to see how these farms are connected. And this is a picture of the cluster A farms, which is by far the biggest cluster of farms. And we look at the period of infection, so the time between diagnosis and the culling of the farms. And we could see that when you look at the sequence of all those farms, that these farms seem to be connected. And we would like to look, and that's what we're doing at the moment, to see how the geographical situation and the time and the risk of infection is connected. So we are developing kernels to see if we can shed more light on the transmission between farms. What you can see in this picture is there seems to be a seizure between 2 parts of that cluster, and that's also something we are focusing on. Is this cluster split, or yes or no, or is it simply a coincidence that there is a sort of seizure halfway this cluster. So when we look at the different parts of that cluster A, we could still see that there is there seems to be a pretty normal relationship between infection hazard and the distance. So it looks like these farms indeed have infected each other in the sequence that was shown in the former slide. But we're still working to see how we can even better fit this in a model. What we're also looking at is what levels of virus these farms have been shedding. And in this picture, you can see of the different clusters, all the CT values of the first throat swabs and secondly, anal swabs to see if there is a link or could be an explanation by the level of virus shedding from the farms in the different clusters. So we're still working on that. This picture is not complete yet, but we're still working to try to shed more light on the risk of transmission between farms. What we're also looking at is the variable, other variables.
I'm going to have to push you to wrap up now, Wim.
Yes, this is the final slide. So what we're also looking at is the days between infection and culling, the farm population, the number of animals in the farms, and also the number of people in the farms. So we are still doing evaluations to see if we can find an explanation of the continuing spread from farm to farm. And that's where I would like to end. Here are all the people that have been working on these outbreaks, and we are still working on papers of these outbreaks. And I would like to thank you for your attention. I will be happy to answer questions.